showing you a set of passage cells. You start off by assessing the cell confluency, then you remove the old media, trypsin the cells in view under a microscope to make sure that the cells haven't become suspended. You want to remove almost all of the trypsin to leave a little film for the suspended cells to be able to migrate. And then you want to tap the flask on the desk and then view it under the microscope to see if the cells have completely detached. Then you want to add the appropriate amount of trypsin to split, and then add 3.5 milliliters into the media with the cells into each new flask.
Alright, we begin passaging by bringing out our cell flask. We know it's ours because we labeled it here in the appropriate place, and we put our date as well, 11/24, in passage 1. It's important to know which passage your cell is at. Oh, and it's also very important that you hold it. You can do anything you want as long as you don't get the media or liquid into the cap. That's contaminated. So we place our cell on the microscope, and we're going to check for confluency. And would you look at that? It is over 70% confluent, meaning we're ready to split.
Alright, now that we're ready to trypsinize our cells, the first thing we need to do is make sure that our hood is sterile. So what we have is we typically have a blower on, but for these purposes we're going to keep it off so that way you guys can hear what I'm saying. So another thing that we do to make sure that it's sterile is we get 70% ethanol, and we make sure to spray it down in the hood and then wipe that down. The 70% ethanol is going to kill whatever bacteria or fungi may be present. And also it's generally a pretty good idea to put some ethanol actually on your hands as well. And you can just rub that in and then put it over this suction area right here. Although it sucks, it'll still help dry your gloves. So the first thing you want to do when trypsinizing is you want to remove the media, because the media will actually deactivate the trypsin. So adding the trypsin to the media, it defeats the purpose. So what you want to do before you start is just loosen the cap a little bit so it makes it easier to take it off when you're ready. Then you're going to get a 5 milliliter pipette, which is sterile, and they sterilize it using gamma radiation. And you're going to just push right here and pop it through. And you want to make sure you want to do it at this end, because if you do it at the tip, it'll cause contamination and it won't be good, so you have to throw it out. So as long as it stays in here, it remains sterile. So just remove it as so, put it off to the side, make sure that your pipette is on, and then using your pinky, grab the lid, making sure it doesn't touch anything on the bottom. So you don't want to touch the lid because that'll cause contamination. And you want to carefully put the pipette in and get it kind of like the fluid in the corner so you can suck it all up. And you want to make sure to stay away from the bottom part of the flask, because that's where the cells are, and you don't want to accidentally knock them off in any way. Now that I've emptied all the contents out of the flask, we're now ready to trypsinize. So what we need to do is get a new pipette tip to keep from cross-contamination. And we want to get our vial of trypsin. So typically we want to take 1 to 1.5 milliliters of trypsin and then add it to the flask. So again, we want to loosen the tips of both so we have easier access to it. And we want to start off with the trypsin. So again, holding the cap with your pinky, you want to take about 1.5 and carefully put it back on. And then now, we could add that to our flask. So what we need to do now is again be careful of getting it onto this surface, because if we get onto that surface we could blow away the cells. So what we want to do is angle it and put it down that way. So that way we didn't knock off any remaining cells. And now we're going to check again on the microscope to make sure none of the cells have knocked off.
Alright, now we're going to check with the microscope to see if our cells are still adherent and they're not moving yet. Alright, good news. They haven't moved off, so we're safe right now. If they did move off, we would centrifuge with the trypsin in it to remove the trypsin so the cells don't die, if they did come off the cell. But since they didn't, we'll be fine right now, and we can remove the trypsin.
Alright, now that we've verified that the trypsin hasn't caused the adherent cells to come off, we're now going to remove the trypsin, leaving a small amount to leave a film for the cells to suspend in, so we can check to see if they're moving around. Alright. See the remainder trypsin right there.
Alright, now we're going to incubate our cells with a little bit of trypsin remaining to activate the trypsin fully. This will allow us to remove the adherent cells into a suspension, which we can then split. So, we're going to our 37-degree incubator. Place it here. And we're going to leave it in here for approximately 10 minutes, and we'll check back on it then. Incubation is passed. We're allowed to check our cells to make sure they're no longer inherent. We're going to do this by doing a quick tap, making sure the cells are up, and we'll check. Would you look at that? There is no layer at the bottom where you can see the cells moving over, so everything is in movement, which means we can now split.
Alright, now that we see that our cells are no longer adherent, we need to add the appropriate media to deactivate the trypsin, because if the trypsin is in there for too long, it could actually kill our cells. So we want to deactivate it as soon as we possibly can. Alright. And last time, it was very important for us to refrain from hitting the back of the flask to remove our cells, but now what we could actually do is spray it against that, because any cells remaining on there we want to knock off. So this time, when we put it in, we could put it in at that angle and to spray it across right here. Alright, now we just want to let the cells mix around a little bit. After we allowed our cells to mix around, we can now take a cell count, but unfortunately, since this is for demonstration purposes only, we unfortunately don't have the cell counter with us, so we can't show you how to properly do that. But what we would typically do is just get 10 microliters of this, mixing it with 10 microliters of the trypan blue, and that will allow us to count the cells that are alive versus the cells that aren't alive, since the trypan blue will actually only dye the dead cells. Then after that, we could get a better representation of how many cells we actually have in here, so we know how many flasks we could split into. So let's just say for these purposes that we are going to split it into 2 flasks. So we already have 3.5 milliliters in this flask, and so we want 3.5 into each. So we're going to add another 3.5, making the total 7, and then put 3.5 into either flask, and then we'll incubate. So again, we're going to change our tips. Allow the media to mix, and then we're only going to need one more pipette tip, and we can actually use one to split into both as long as we remain sterile while doing so. So what we're going to want to do is set up both and make sure we loosen the cap like we did before to give it some easier access. Alright. So what we're going to do now is remove 3.5 out of here, and then transfer it to the first flask. And as we do that, we're going to remain hovering above the flask, which will keep this tip sterile. It will not create cross-contamination. So now we could put this tip back into the original flask, so we could take out the remainder of the media and cells, and add that to the new flask and not worry about contamination.
by making sure that we label both our flasks so we don't get confused. Today we're going to put the date of 11/26 and our passage number, which is now passage 2, since we began with passage 1. We're also going to put our group symbol, an M and an R, so we can distinguish between other groups. We also want to put the name of the cell that we're going to be using. In this instance, MEFs, mice embryonic fibroblast cells. And this can also be done on the second flask. 11/26, passage 2, MEF, and group name. We will now be taking these to incubate. Alright, we're back in the incubation room now, and we're going to be putting the same 37-degree incubator that we used when we trypsinized. Place them in a place that we'll remember. You can stack them if you want. Save time, doesn't matter. Leave them here. And we're going to leave them overnight, and they'll stay there until we're ready to use them again.